Hello, I hope everybody's doing okay with this lockdown, such strange times. What we're going to cover, so I'll give you guys a background on some statistics for your FHS paper 3, basically, is the way we used to usually run this when everybody's live in person. I'm going to try to do a live event with everyone on Zoom on Friday, so hopefully you can make that. I'm also going to try to record the Zoom meeting on Friday, so if you can't make it live, you'll be able to see what we discussed during that meeting. With this particular seminar, I guess, for lack of a better term, what I'm doing with you is an FHS statistical seminar. Over this, I'm going to cover a little bit of theory. I'm going to mostly try to make it very pragmatic, so it's just what to look for in a paper for your FHS exam. And it's beyond that. It's what to think about in terms of statistics in general, what you need to know. Some people get too bogged down in a lot of different aspects of it. Hopefully this you can take away for even after you're done the FHS, be able to look at a paper quite quickly and critically and see if the statistics are appropriate. What I'm trying to do in this particular little video is to just run through some of the principles that we will then go into and discuss if the live version works. This is not on Panopto. I was trying for the last 5 days to figure out how to use Panopto with a whiteboard. I'm going to sketch things out on an iPad. So this was working earlier in some of the tests. It's the only way I can do this with Zoom. So I guess it's a test of Zoom as well. And Zoom seems to be better than the Microsoft Teams product at this point, or Panopto, where I can't do this. I feel very constrained here. Usually this is very interactive. So I'll go through things. On Friday, you can ask me in more detail if something doesn't make sense. I can't cover all these stats. This will probably be about an hour. I can't cover all stats in an hour, and I might go off on tangents and this sort of thing. So I'll try to stay focused. We'll see how that goes. Right now, I'm just going to share the screen of my iPad. And because this isn't Panopto, I don't think I can edit any of this stuff out. So there's going to be little moments where you see behind the scenes. Okay, looks like it's working. I'll have to take a chance later that this is actually working. So the document, I put some documents up on WebLearn, and then Rachel moved them into wherever you guys are going for WebLearn stuff now. So hopefully you'll see it there. I've got another computer screen over here. So this is multi-screen, multimedia stuff. This one I've got up a document saying FHS signaling statistics rough guide. So this is like the rough guide that I put up on the internet, on the WebLearn. Hopefully you can find that document. And it's just a very short document going through all the different things that I think you should look at for statistics. Before I get into it, I often get on a soapbox on this, so I apologize, but I've got to get it off my chest, otherwise it'll be in the back of my mind through this whole thing. The statistics that we teach you, the statistics that are used by 99% of all biologists, medics, biomedical scientists, these types of things, are what we call frequentist statistics. They deal with statistics where you have probabilities. And what they have done is, if you have a certain outcome, you know what the expected outcome is, and then you say, if we did this experiment an infinite number of times, what would that look like? So if we're dealing with a normal distribution like this is the mean, this is 1, 2, 3, these are 1, 2, 3 standard deviations. It tells us the central point and the spread, or variation, or variance, or scatter. So with 2 points, we can look at this curve. Most of biological statistics deal with this, and it's called frequentist. And like I said, it's if you did the experiment an infinite number of times, this is the population that you would get. And then we ask the question, if we're observing differences in height, let's say, between two different groups, is the difference in height what we would expect in, is it 1 standard deviation, 2 standard deviations, 3, 4, 5, from what we would expect from this theoretical population? And then we can take the area under this curve. So area is equivalent to probability. So we call this frequentist probability-derived statistics, and this will mostly be with things that you'll be familiar with. Normal curve, which is what I sketched up here. There will also be the tests you're used to. T-tests and Anova are based on frequentist statistics. T-tests are not based on normal, they're just based on a T distribution. And I'll say a little bit about that later. All of this, like I keep emphasizing, is one way of thinking about statistics. From all of this, the P, very often people say P has to be less than or equal to 0.05, or your result is not statistically significant. There's a hard cutoff, there's a cliff edge. I've taken statistics like this since I was in grad school. In Canada, I was in a program where we did crop science, and I took a lot of graduate statistics. It's very appropriate for doing those sorts of things. Whether it's appropriate for doing lab-based statistics, whether it's appropriate for doing other types of statistics, the jury's out. This frequentist is also called Fisherian because Roland Fisher came up with some of these techniques. The t-test is from a guy called Gossett, William Gossett. William Gossett's better known by his pseudonym, Student. So it's sometimes called the student t-test. I might have a little time to talk about that later. So all of this is frequentist statistics. However, there's a whole different field of statistics, and these two compete with each other, and that's called Bayesian. And it comes from a guy called Bayes, who was, I think, some clergy guy. And I think these Bayesian statistics make a lot more sense. However, all of biology is fundamentally on frequentist statistics. So that's the way we're going to end up discussing this. There are huge advantages with Bayesian statistics. I don't have time to go into them now, but now and then that will leak out. What I'm going to focus in on now is all frequentist statistics that your FHS will be about. But once you're done with this stuff, and in the rest of your life, keep an eye on the Bayesian statistics. That's the future. I think it makes a lot more sense. It's more intuitive. So what I'd like to cover now is a few things from this FHS rough guide to statistics that I put together. And it's mostly just my opinions. I've backed them up by references. Statistics at the cutting edge, experts disagree, all of this. I might have time to say a little bit about that later as well, about even the origins of these frequentist statistics are a bit of a bastardization of students' ideas, Fisher's ideas, Neyman Pearson's ideas. Neyman Pearson really didn't get along well with Fisher. They had huge fights about this. Somebody wrote a textbook and combined their two ideas, and they're incompatible. Yet that's the way everybody does statistics now. But I'll try to not say too much about that. I'll try to work just through the system that we're given right now. So I already highlighted that we have a normal distribution, like something like this. And if we have a mean, let's look at height, and this is roughly true. It's not quite, but it'll get the point across. Let's say the average height is 170 centimeters of a person, and the standard deviation, so the distance that the data spreads, is 10. So we'll say the standard deviation, this is a population. With populations, we have parameters. So think P for population, P for parameter. The standard deviation, which is the spread of the data, we use the Greek letter sigma. That's the spread of the population if we took everybody's height in the world. So there's what, like 6 to 7 million people, billion people in the world. If we measured absolutely everyone, we would get the standard deviation and the mean, the central tendency. Because this is a population, we use a Greek letter, so mu. This is mean, and sigma is the standard deviation. If we're looking at height, the real height is probably about 175 centimeters, but I can't do it in my head. So let's say that the standard deviation is 10 centimeters. So then we would have 180, 190, 200, and then 160, 150, 140, 1, 2, 3. So each one of these is a standard deviation. It's the spread. With these two values, we can fully describe a normal distribution. Then we can say if somebody is greater than, let's say, 190, if their height's over 190, then are they taller than would be expected? So with this particular normal curve, we don't keep the numbers. What we do is we switch the numbers into a common curve so that we can use similar lookup tables to know the probability. The probability is area under this. So we transform them. So let's say an example here is we would transform this to a Z. This this particular distribution, we use a Z statistic. And what we do, let's say we had a value of 190, subtract 170. So this is the individual you're looking at, X1, subtract X bar, which is the mean of the population. And then we divide it. We want to normalize it. So we divide it by the standard deviation, which in this case is 10. All of this is in centimeters, but it doesn't matter. So this would be X1 subtract X bar divided by sigma, we'll put as the standard deviation. And I'll actually put mu there because we're dealing with populations. At this point, then we would see that the difference between 190 and 170 is 20 divided by 10. This is 2. So our standard deviation for, put another line in here, and I can't seem to get my eraser. Anyway, this will be 2 standard deviations then. 180 would be 1. 0 is the mean when you do a normal curve like this. Then we have -1, -2, -3, and then +3. So this is normalized. These are Z values. So once you get Z values, you can take any data set and convert them to these. The problem is for a population, as I said, this would include everyone on the Earth. So I can't remember, it's like, I think 6 to 7 billion. We can't do that. So what we do is we sample. All of statistics are dealing with sampling. So rather than having the known population that only exists in the guts of a computer, what we do is a sample. So we go into this population and we pull out a group. We might pull out 3 people from that, measure all their heights. all of them would have a certain variation, but then we would average their heights. So if we pull out 3 and we get a certain height, so now this is going to be our sample, and the sample has statistics. So remember, sample S, statistics, population P parameters. We will get, if we keep taking, let's say, a sample of 3 and just keep repeating that, we will get what looks to be sort of a normal distribution, but you'll notice it's broader. So what we're doing here is we're in a group of people, the population, we take 3 people, we measure their heights, we average them, and we plot that. So we plot that. That would be one number under this distribution. Then we put those 3 people back and we randomly select 3 more. And we keep randomly selecting 3, taking that average, the mean, and plotting it in this distribution, the sample distribution. What we end up getting is, if we were doing 3, this would be n equals 3. You see that it's broader and the top is squished down. This is a slightly different distribution. This is our so-called T distribution. And it comes from small sample sizes. This is, the distribution's shape depends on the number of sample sizes. So if it's 3, it's really broad. If it's 2, it's even broader. 1, you can't get a distribution at all. Then as you go up to, if you went up to 10, it will start looking like this. So it starts squishing in. And then once you're up to about 50, so this will be 50, this will be n equals 10, n equals 50. Once you're up about 50, it looks just like our standard normal distribution. It looks just like the normal distribution. So T distributions, T tests, are useful for small sample sizes. Anything under 50, anything under 30, you probably should be using a T distribution rather than a Z distribution. Just an aside as to where this came from. This was actually a trade secret back in the day. This came from Gossett, this William Gossett that I mentioned earlier. So William Gossett, well, there's an Oxford connection with this. He was actually a chemist reading chemistry at New College. And once he finished with a first-class degree, the brewing companies, Guinness and a few others, were buying up Oxford graduates at that point. And Gossett went to work for Guinness in Dublin, and they had a problem comparing different plots of barley. This is not where you could do an infinite number of experiments and use statistics. That was the preserve of theoretical mathematicians who really dominated statistics back in the day. Gossett needed an actual useful statistical procedure to look at different barley varieties. So you get barley, you take the grains, you germinate them, you dry them down, that gives you malt. Germination converts all the starch into sugars, so you get these little barley like kernels that are just pure sugar. Then you use yeast, it converts it into alcohol, and you add some hops and you get beer. Guinness had concerns about how much hops they should use, what the effect of protein was on this. So he needed small sample statistics. He came up with the T distribution for small sample statistics, where the shape of the curve is dependent on the number of samples that you take. You don't know the population, but then you can estimate the population parameters. You can estimate the mean and the standard deviation from your sample. That's statistics in a nutshell. Gossett, as I said, wrote under the name student. He wrote under the name student because his employers didn't want him to publish because it was a trade secret. Nobody wanted to reveal statistics like this. So everybody finds statistics a bit of a pain in the butt, whereas at that point, this was around 1900, statistics were a very valuable trade secret. So perhaps today on this video recording, the Chinese and the Iranians and everybody and the Russians are all trying to listen in on UK scientists to steal data about our immunization bits. If they get this, these are pretty old trade secrets, probably not going to help them that much. In terms of this being a sample statistic, we don't call it mu and sigma because it's a statistic. We use Roman letters, and the mean, the central tendency, is x bar, and the standard deviation is SD. So they mean the same thing. There's still a mean and a standard deviation, but because they're statistics, we don't use the Latin letters. So if you see Greek letters, it's population. If you see Latin letters, it's not the population. The population can be anything you want. Could be the average height of all the students listening to this particular video. Could be the height of all the FHS students. Your population could be all the medical and biomedical students. So you can sometimes know a population. If you're watching the news over the COVID stuff, everybody's fascinated about this R, and R is given as 0.8 plus or minus 0.6. Those are confidence intervals. We'll touch on those in a little bit as well. My take home up to this point is statistics are sampling based. We know what that distribution looks like. Then we can use some of the principles behind that to ask questions. If we sample from two groups and they're going to be different, unless you're really, really lucky and they completely overlap, they'll be different. Then we ask, is that difference? What's the probability of that getting that difference or more based on these curves? And there's a t distribution that mostly we'll use. It's based on the standard distribution, normal curve, but it's spread out if there's low numbers, and it will start resembling a normal distribution if you have high numbers. So I'll now move on a little bit. Mostly what we want to ask is let's say that we measure all the medics and all the biomedical scientists, and we measure height, and we get a number here. Let's say that we get 170, and over here we get 180 centimeters. And we can ask the question, are these two different? So then we need to know, what was our n? If n was equal to 3, that's going to make a big difference compared to n equal to maybe 20. Because remember, our t distribution depends on that. How do we look for a difference? There's an equation, a t-test, and this is, it's useful learning this. So the t-test is, so t, and it's usually at a certain percentage. So we'll say it's 0.05 is equal to x1. So the mean of, let's say, x sub m, the mean of the medics subtract the mean of the BMS students. And then we'll divide that by, this is where it gets a little weird, and I'll try to explain this. I try a different explanation every year, and it never seems to work, so I'm still working on it. You guys are a new experiment this year. We'll divide this by SEM, standard error of the mean. So if we think about what this means, we've got our t distribution, and from this t distribution, we'll have a mean, and then we also, it looks like a normal curve. So we have standard deviations, but these are not Z scores, they're T scores, but they're still standard deviation, meaning if you have one standard deviation from 1 to -1, you will get, like 67% of the population. If you do 2 standard deviations, it's 95%. It's just that these are spread out more. You need a higher T to get that percentage. So with going back to our equation up here, we will subtract, let's see, that would be 170 subtract 180 divided by standard error of the mean. Standard error of the mean, we would actually derive from the sample population. Remember, we don't know what the real population is, so we'll derive it from the sample. Standard error of the mean is equal to standard deviation divided by the square root of n. So you can see that as n gets larger, our standard error of the mean, so as n increases, standard error of the mean decreases. And we're dividing, in our equation up here, we're dividing everything by the standard error of the mean. So our number is going to get smaller and smaller. Our t value is basically a z value, but based on the t distribution. So if we can cheat a little bit, and we know that the standard deviation of a population is 10. So let's just see what that does. Let's go standard error of the mean is equal to 10 divided square root. Rather than 3, let's go to 4. Then I can do the square root. So square root of 4, that's 2. So 10 divided by 2. Our standard error of the mean is 5. Now let's think about, so if we plug that into the equation, our t equals 0.05 is equal to 170, subtract 180 in centimeters, divided by standard error of the mean, which is, in this case, 10 divided by square root 4, which is actually 5, and the top is 10. So 10 divided by 5 is 2. So this t is what we determine the difference, how far apart the two samples are. They're two standard deviations. So then we can construct a t distribution with differences. The way you do that is you have the same mean. So in terms of the Greek, so it's a population, and then we sample. We know that these two curves are identical populations, so you just keep pulling values from a population. And from that, you end up getting a curve that's much tighter than the population. And this is differences. So what you do is you pull values from one population, pull values from another population, subtract them. You'll get a difference because you're randomly pulling different people. This difference is just due to sampling. So it's sampling error. So that's why we call it standard error of the mean. Here we're getting a curve of means. And the higher your sample number, the narrower this is. So getting back to our example, if our n was 4 up here, we got a t value of 2 down here. Sorry, I'm realizing I can't point at this screen. If I point at it, you guys can't see it. One deficiency, I'll try to work on that. So this t value of 2 means that we're up here. What's the probability of getting a t value of 2? Is that significant? If this was a z test, it would be. Our critical t is 1.96 on a z test. On this one, we'd have to look up what the t value is, critical t, for 4. Off the top of my head, I think it's around probably 3 to 4. So 1, 2, 3, 4 on our t graph now down here. You'll see that 2 is less than 4. So we would conclude that there's no statistically significant difference between the heights of the medics and the heights of the biomedical sciences. If, however, let's plug in our numbers for n equals 20. So then we've got standard error of the mean is equal to 10. Remember, we wouldn't necessarily know that. We can calculate standard error from the sample, but this is just an example. And then we'll divide that by the square root of 20. I don't know what the square root of 20 is, but let's see. 4 times 4 is 16. 5 times 5 is 25. Let's call it 5. I know that that's 25, but let's just call it that. So 10 divided by 5, wait a second here, is 2. Sorry, I made a mistake earlier. Our critical t before should have been 5. So, excuse me. Our critical t, or sorry, the sample t, if there's a sample size of 20, is 2. If the sample size was only 4, our t value was 5. And that relates to this normal distribution or t distribution and how many standard deviations are we away from the mean. So you notice as our sample size goes up, we need fewer, a smaller distance. We get more sensitivity, more power to detect real differences. So that's the basis of a t-test. So a t-test, just to reiterate, it's difference between two means. So x1 subtract x2 divided by the standard error of the mean. Standard error of the mean, I don't think I did a good job explaining that. So if we have a population, and let's say the mean is 170, its height. Let's sample. And we sample n equals 10. What you will do then is each one of those will be, if this is 170, you will get a value, 60, 180. You'll just get a curve. And eventually you'll do 10, then you'll do another sample of 10, then you'll do another sample of 10. And each one of those means you plot. What happens is, because each one of those samples is going to be closer to the real mean of the population, this curve is going to be narrower, but it still has the properties of a standard curve. So we still have a mean and we have a standard deviation. The standard deviation of this sampling means sampling curve is called the standard error of the mean. So as you increase sample number, this shrinks. As you increase sample number, that t curve starts to narrow and resemble normal distribution. If you guys don't like the theory behind this, you don't have to know any of this. Now I'm going to get into some more rough and ready things. Oops. For FHS stuff, which you guys are probably more interested in. This is very pragmatic. So in terms, looking at my cheat sheet now, what do you look for in a paper? Error bars. So there's usually error bars, but they can mean very different things. So always look in the legend. Hopefully in all your reports of your lab projects, in the figure legend you gave n, what the error bars mean, and what statistical test you used. In terms of error bars, we'll look at a couple things here. Let's take 3 values first. So we could have with 3 values, so this is, let's stick with height. And down here, I'll have different n's along the bottom. So n is equal to 3. This could be range. So we could just take the top and bottom and say that's range. Those could be error bars. Other error bars that you will see could be standard deviation. So the standard deviation, I won't derive that right now, but it's that measure of spread. So standard deviation is going to be smaller than the range. So let's just say standard deviations like this. Then we also could have standard error of the mean. What I want you guys to do right now is think about what a standard error of the mean could be here. Is it going to be bigger, smaller? How's that going to be? For your exams, memorize the definition of the equation definition of a standard error of the mean. So just to reiterate, standard error of the mean, I'm writing it down now, is equal to standard deviation by square root of n. So the square root of 3, I don't know, it's like 1.6, something like that. So we would divide the standard 3, our standard deviation, well, we've got to put a number to our standard deviation, but we would divide it then by the square root of 3, which is 1.6. So it's going to be 1.6 times smaller than, it's going to be your standard deviation divided by 1.6. I guess that's the easy way of saying it. So we'll have a mean. The mean won't change, but our standard error of the mean is going to be only 1.6 times as large as the standard deviation. What about, well, yeah, there's also something called confidence intervals. They're probably the best thing to use. Everybody argues about whether you should use standard error, standard deviation. A lot of people say standard deviation. Standard deviation is fixed for the population. Let's jump way out here to the right and say, what if n was 100? And so now we'll have more dots. The range will actually probably be wider because there's more of a chance of getting extreme values. So this will be our range. The median, the mean, is not going to change. The mean will be the same. You might get some differences in it, but that's just sampling error. The mean is going to be the same. So these lines here are means that I'm drawing. Standard deviation, think about this. Does your standard, so mean doesn't change as you increase sample size. Does the standard deviation increase as you increase sample size? You might want to think about that a little bit. It actually doesn't. You get a better estimate of it as you increase sample size, but it doesn't change. So our standard deviation is going to be within the error of the measurements, the exact same for n equals 100 as it is for n equals 3. N equals 100 will be a more precise measurement. What about standard error of the mean? We know from this equation that I just showed you that standard error of the mean decreases as n increases. So if we do this for n equals 3, we said that it would decrease by root of 3, which is equal to, I don't know, 1.6, we're saying. You can look up the real value. If n equals 100, I chose that because I can do the square root. Square root of 100 is equal to 10. Your standard error of the mean will be only 1/10 of the standard deviation if you're using n equals 100. So on this one, I can't even draw that in. I've drawn a little bit here, but it's an exaggeration. It would be so small you wouldn't even see it. Think about the extreme. Think like a calculus person. What's the limit? If our sample number goes to infinity, or more realistically, goes to the actual population size, you know all of them, so there's going to be no standard error of the mean. What you measure is the real mean. So low numbers, we're estimating the mean. Higher numbers, we're getting closer and closer to what that real mean is. So the standard error of the mean is a value that tells us how well we know the mean. It's called an inferential statistics because we can make inferences from it. We can judge different things from it. We use the standard error of the mean in the t-test to ask how many standard deviations are these differences apart. So the standard error of the mean is a derived number. It's a statistic. It's an inferential statistic. The mean and the standard deviation are what are called descriptive statistics. They do not change with the population. They are what they are. They never change. In terms, I shouldn't probably do that. I'm not sure what is recording, but I was just trying to move some of the stuff around. In terms of what I was saying, if you're asked on an exam, why would somebody plot standard deviation? Why would they plot standard error of the mean? The best answer, probably as far as the examiners are concerned, we'll talk about some of the examiner comments on Friday, I would think is a standard deviation is a descriptive statistic. They want to show the spread of the data, the variability in the data, the variance of the data. Standard error of the mean is if they're doing inferential statistics and they want to show you how well they've estimated the mean, they'll use standard error of the mean. Right now, I'll introduce another concept. So remember, standard deviation doesn't change, mean doesn't change, they're fixed in a population. The statistics are trying to estimate those values. I'm going to now introduce something called confidence interval at the bottom here. So confidence interval is equal to the mean of the sample plus minus, so it's an interval. Rather than a point estimate, we're giving an interval. Plus minus the standard error of the mean, and then you multiply it by t at 0.05 if you're using 0.05 as a statistic. So some of this looks very familiar to the t-test. We're using it, that's where we calculate a t. Here we're saying we're not looking at the differences to calculate a t. We're just saying what's the t at a given value. Remember, t is the critical t, where there's 95% of the values included and 2% on one side and 2% on the other side. So it's a two-tailed t-test. That critical value depends on the sample size. So t is going to change. So if we put t on our chart up here, t for 3, like I say, I think it's around, let's call it 4 for sake of argument. T for 100 would be probably 2.01, something like that. It's getting damn close to that 1.96 for a Z distribution. So our critical T in our confidence interval is going to depend on the sample size. So for a confidence interval, if we're using, n equals 3, so if we go back here, our T is 4. So if we plug that into our standard error, our confidence interval equation, we have to take the mean times standard error of the mean plus standard error of the mean times the T. Our standard error of the mean we can get just off our graph here, and we have to multiply that by 4. So here's our mean. There's 1, 2, 3, 4. Then we do the other side, 1, 2, 3, 4. So it's the confidence interval is 4 times the standard error of the mean. Confidence intervals are useful in terms, if there are differences between the means. If I use the example where we were comparing heights between the medical students and the biomedical sciences students, and we get differences. So we take 170, subtract 190, and we get a difference of 20. Then we divide it by the standard error of the mean. If those differences, then we can calculate confidence intervals. If our confidence interval doesn't include 0, we know that 95% of the time, the confidence interval won't include 0. If you're looking at differences, if it includes 0, that means there's no difference. If it doesn't include 0, there's a difference. So that's why confidence intervals are quite nice. If you're graphing with standard error of the means, the standard error of the mean will change depending on your n, whereas a confidence interval, it's locked in. If it includes 0, there's no difference. If it doesn't include 0, there's a difference. So they're much preferred. The reason people don't plot them is just look at the graph here. They get horribly big. We've tried to publish data with confidence intervals, and we've gotten beaten back because people say the error bars look too large. They do look big, but people just aren't used to it. So on exams, probably put confidence intervals are probably one of the best things. They're easy to interpret. They're more intuitive. Let's do the confidence interval for n equals 100 now. So there we said that we've got standard error of the mean would be very, very small. And then we multiply it by a t, which is 2. So we've got a very, very small standard error of the mean, and we've got a t that's around 2. You can't get t lower than 2. That's the standard normal distribution. That's your Z distribution. T approaches 2 as n goes to infinity. So drawing on our graph here, this is an over-exaggeration, but it's 2. You'll notice that the confidence interval for n equals 100 is really small. because the standard error is really small, and also the t-value, the critical t-value, you can look these up in tables, for n equals 100 is close to 2. Whereas if we're doing this for n equals 3, our t-value is large, it's around 4. And also our standard error, the mean, or sorry, our standard error, the mean is quite large because we're dividing our standard deviation by the square root of n. You could do all the ones in between here. The take-home message is that the mean stays the same. So let's say this is the mean. The mean stays the same. Our statistics, because we're sampling, might be around that mean, but it doesn't mean that the population's means change. Also, the standard deviation doesn't change with sample size. The other thing is standard error of the mean shrinks with sample size. As sample size increases, standard error of the mean increases till you hit n equals a population. Max bringing up notices. There. I don't know if this gets taped on Zoom or not. Anyway, so the standard deviation stays the same, mean stays the same, standard error of the mean shrinks, confidence intervals shrink as your n increases as well. So sample size is your friend. As you increase sample size, you get a better t. So now that I've gone through all the different possibilities, standard deviation, standard error of the mean, and confidence interval, I've also covered t-test. If you guys know that, that's all the background you kind of have to need for this. In terms of a graph on a paper, if you're doing your exam or just reading a scientific paper, look at the error bars, see what they mean. Then you can interpret them. If you don't know what they mean, it's very difficult to interpret the data. Yeah, there's assumptions that have to go behind this. They're independent means, not subsamples. An assumption for the t-test is that the data are normally distributed. So this is where I disagree with the examiner's interpretation and a lot of scientists. T-statistics are for small samples. It takes into account that you have n of 3. You can't tell if 3 points are normally distributed. There are normality tests. They only start to work once you're above 10 or 20. Once you're above 10 or 20, there's a concept called central limit theory that kicks in, and you start getting that nice normal distribution that I showed you earlier. If you sample from a population, you get a population distribution of the means. So by the time your normality tests start to actually work, you know that you're already normal because of the central limit theory. But on your exams, if they haven't done a normality test and they're using t-tests or Anovas, criticize them for not using a normality test. However, as I said, these tests t-tests do not require normality. The clue is in the word. It's an assumption. You assume normality. I might as well say, if you keep doing those sample distributions of means, even if you have a binomial distribution and you add things up, or really skewed wonky distribution, look on Wikipedia's got a nice article on central tendencies, and you'll see that even wonky distributions, if you keep sampling from it, you'll get a normal distribution. That's the theory behind frequentist statistics. That's why this stuff works. So if the data aren't normal though, and you're pretty sure they're not normal, you've got enough points and they look really skewed. Usually it's skewed distributions one way or another. First thing you do is you try to transform them. You do a log transform, square transform, square root transform, arc, cos, any of the trig functions. Try to get a normal distribution. If that doesn't work, then you go to non-parametrics, where there isn't an underlying assumption about the distribution. They do not work if you have n less than 6. You'll never be able to get statistical significance with a 0.05 if n is less than 6. Think about flipping a coin. First time's 50-50, 2 coins at once. The chance of getting 2 coins heads or 2 coins tails is 0.5 times 0.5, so 0.25. Keep working that up. You don't crack 0.05 until you get 6 coins flipping at once. So the non-parametrics don't assume a distribution of the data, but you need over 6 before they will, even if all your coins hit heads, you'll only get a significant difference if you have n over 6. Is the test appropriate? Think this, T for 2. It's a bit hokey, but t-test if there's 2 values. If there's over 2, you need to do an Anova. This is an analysis of variance. It looks at the spread of data. It looks at something called variance. I wasn't going to get into this, but I'll say it just briefly. If you have the standard deviation and you square it, you'll get variance. If you take the square root of each of those, standard deviation is equal to, whoops. This is the problem with doing things on the fly. Standard deviation is equal to the square root of variance. And variance is squared terms. So imagine we had our height example earlier. Variance would be in centimeters squared. So it's hard to think about height in centimeters squared. So we take the square root of that, and that gives us standard deviation in just centimeters. But variance is this difference from, if you had 170 and 190, as I said earlier, the difference is 20. The average is 170. So 20, well, yeah, it would be 20 squared, which would be equal to 400. That would be our variance. For Anova, it's variance of one group over the variance of the population, sort of. I don't have time to get into the whole thing. You'll end up getting a ratio between the variances. You can imagine if this variance was really big and this one was small, the statistic here, the ratio statistic, is an F. You get an F test. So Anova looks at the variance. So you can imagine if the variance between the two groups was huge versus the variance within the groups, that's what the new denominator here is, you'll get a big F. A big F means that you have statistical significance. F has a distribution. It depends on the number of samples. We don't have to worry about that much. The computer will take care of that. So getting back to where I was, T for 2, if you got comparing 2 means, use a t-test. More than that, Anova. And Anova will spit out just significant or not, and it will give you some value, 0.001. So that's less than 0.05. You might be able to get 0.049 or 0.051. This is why I hate the idea of frequentist statistics with cutoffs. This would be significant. This would be not significant. That's craziness. These are both on the same sort of ballpark. There shouldn't be hard cuts with this. Yeah, I'm self-censoring. I could go on with that a lot. It's crazy religious type dogma that we use 0.05. Anyway, an Anova will give you a statistic. That just tells you, let's say that we're comparing 6 things, and they all have error bars, probably standard error, the mean, because that's what scientists like to put on their graphs because they don't look big and bad. So if we've got 6 samples here, Anova will just say something in there, bag of samples, don't know what, something in there is going to be statistically significant. What we really want to know are the differences between the means. The way we can go about that is through something called a post hoc test. Post hoc tests, you'll come up with Dunnett, Bonferroni is the easiest to understand. If you're doing 6 of these and you say your statistical significance, actually let's do 5. Let's get rid of number 6. If your critical P is equal to 0.05, that's if even though I don't agree with it, that's what everybody else does. It's significant or not at 0.05, and you do 5 comparisons in our Anova here. So we compare 1 to 2, 1 to 3, 1 to 4, excuse me, 2 to 3, 2 to 4, 2 to 5, all possible pairwise comparisons. What you would do is you would divide your 0.05 by 5, divide by the n, and then your critical p-value is 0.01. So you need a lower p-value before you can declare something significant. This is for multiple comparisons. It basically sets p to 0.05, meaning that you will see this type of difference or larger 5 times out of 100, once in 20. If you start comparing everything, it gets like a coin toss where you just start looking at everything and you're going to be out in those 2 tails of a normal distribution, declaring things significant where it's just chance. Even a normal distribution has values beyond that. Statistical testing says if I take 1 sample, what's the probability of finding it out there? If I took hundreds of samples, I'll be able to find ones that are out there, and you get spurious conclusions. So make sure that they've done a Bonferroni correction or a Tukey. Tukey's is another one that they could correct for. Yeah, I've said a lot of this stuff. I'm just going through my notes right now. Said a lot of this already. T-test Anova. There's paired t-tests. So you can imagine if you were doing blood pressure and you've got a drug to lower blood pressure, but some person comes in and they're at 110, another person's at 130, another person's at 145, another person's at 160 systolic millimeters of mercury. So the drug, let's say the drug decreases pressure by 5 millimeters of mercury. It's probably a good drug. That's not bad. But if you just do all the means using the raw values, it's going to be off. So what you very often do is a baseline value in one person and then subtract their later tests. This is called a repeated measures design or a paired t-test. If you're using Anova, it's repeated measures where one candidate you follow a long time. T-tests, same person at 2 different places. I've mentioned multiple comparisons. I've already bitched about the p-values. Power is, so your ability to detect a difference. How should I put this? So the null hypothesis statistical test, so that's basically, does it crack 0.05? You can change which statistic, what your critical value is, but that's the most common one. If the null hypothesis statistical test, all of this, there's a lot of philosophical things about your testing where you assume that there's no difference and then you get evidence to disprove that and all that. It's hypothetical crap. If you want to read about that, feel free to. We can talk about that in the live session too. Right now, what I'd like to emphasize is the difference, whether you declare something different, is due to an effect size, and it's also due to square root of n, or the sample size. So if you remember the t-test, it was equal to a difference in 2 means divided by standard error of the mean. And standard error of the mean, you should be really bored of me writing these things out. You should say, I already know that. Make sure you memorize it for the exam. Standard deviation divided by root n. So as you increase n, you're going to shrink your standard error of mean until it gets so small that you'll be able to declare anything statistically significant. A lot of these studies where they use thousands or 10,000s or hundreds of thousands of people, the effect size, this thing, our blood pressure, millimeters of mercury, they might be able to detect a 0.000001 change in millimeter mercury, and it could be highly statistically significant. So your p-value could be 0.000001, and millimeters of mercury, the difference could be 0.000001, and it would be statistically significant. But the crucial thing here is it clinically significant? Is it practically significant? So effect size is really important. If you just keep cranking on the replicates, you'll make anything statistically significant. This is another reason why I don't like current frequentist statistics. They focus too much on statistical probability, the actual probability, the p-value, and they ignore effect sizes. Effect size is really important. Is this worthwhile pursuing or not? Replicates. Imagine you have looks more like a rat, but it's supposed to be a mouse. You have a knockout mouse, and you have a gene that affects the length of its tail. Now, imagine if you measured that tail 10 times, could you do statistical testing on it? Probably not. The replicate, the biological replicate, is the mouse itself. Doesn't matter how many times you measure the tail. So if somebody has a knockout mouse and they take cells from it and they measure the cells 10 times under a microscope, your biological replicate's just 1. With cell cultures, this gets really complicated and people argue about it. With a Langendorf heart, if you have a heart up beating, not Langendorf, if you take a heart and you disperse all the cells and then you look at them under a microscope, so you're looking at cardiac ventricular cells, is each cell n equals 1, or is each cell, if you measure 10 measurements, are they just subsamples? And the guinea pig very often that you took the heart from, is that your replicate? People argue about these things. Be familiar with the arguments. What is your n referring to? What's the actual biological replicate? Said all that. So yeah, I've gone through everything now. I hope some of that was at least clear. It's much better when it's interactive because I don't know if I've missed something important and completely confused people. There could be errors in this as well because I read around and I did it on the fly. So if there's errors, call me up on it on Friday. Hopefully this Zoom thing worked, and hopefully the Zoom thing works on Friday. So there are exercises. Hopefully you've looked at that on WebLearn. So go through there. There are past questions from paper 3, and we'll use everything that I talked about here to talk about how I think it should be. And of course, I'm right. Everybody thinks they're right. And then we'll talk about how to answer it from the FHS perspective. And we'll also talk about how you guys maybe should be thinking about some Bayesian statistics, not for the exam, but for just your edification when you leave this place. Okay, hope to see you on Friday.